So here we're going to focus a little bit more with once those cells have received the communication from another cell, what they do with it, how they process it, how they change because of it, okay, how that communication can impact the cell. So we're going to look at that in a little bit more detail. So looking at our big picture here so that we don't lose sight of it, okay, this whole thing is called our signal transduction pathway. And that signal transduction pathway is basically the pathway that's going to link the reception of the signal to the cellular response. So it's going to link, sorry about that. It's going to link reception over here to response. Okay, and so the signal transduction pathway is going to have kind of three basic parts: the reception, the transduction, which is going to include a lot of changes within the cell, modifications to proteins, phosphorylating of proteins, and then the actual final response of the cell. And we're going to look at each one of these parts in detail. Okay, so for example, if I have a surge in epinephrine, so adrenaline hormone, okay, there's a change in the environment, I have the surge in epinephrine, that's going to cause the cell to break down glycogen. Glycogen is how our cells will store glucose. So the final response is going to be that release of sugar so that we can help fuel cell respiration, so that we can do our fight or flight response. So the signal transduction is looking at that pathway from that stimulus, that surge in epinephrine hormone, to that final response. How does it get there to have a response of actually releasing the glucose? So let's start with looking at the reception part of this cell signaling pathway first. So the reception is going to be exactly what it sounds like, the actual binding of the chemical messenger to the receptor. So the chemical messenger is a ligand. It's spelled for you right there. So the chemical messenger is a ligand, and the ligand it could be a protein-based ligand or it can be a steroid-based ligand. So based on the characteristics of the reception, you can tell which kind it is. You can tell if it's steroid-based or if it is protein-based. So the receptors for a protein-based ligand or a peptide-based ligand, remember proteins are also peptides. So the receptor for a peptide or protein-based ligand is going to be on the outside of the cell, on the cell surface. You can see that in this picture right here. There's our cell membrane in gray. Right? We've got our extracellular fluid in blue, the cytoplasm in yellow, and this is my receptor, that big huge purple molecule there. That's my receptor. And so that receptor for a peptide or a protein-based molecule is on the cell surface, because the ligand, the protein-based ligand, cannot cross that phospholipid bilayer. Remember I told you all these things are going to be connected and we're going to keep pulling them back in. So if we think back to cell membranes, right? The cell membrane is made of a phospholipid bilayer, has that big hydrophobic region, so it doesn't let those proteins cross easily. So a protein peptide-based ligand, it can't go into the cell. So it has to bind to a receptor on the cell surface, and then that triggers changes within the cell. Steroid or lipid-based ligands, their receptor is going to be inside of the cell, okay? Because a steroid or a lipid-based ligand can cross through those phospholipids. And so their receptors will be all the way inside of the cell, usually all the way inside of the nucleus. And so you can tell pretty quickly based on a diagram if it's a protein or a steroid-based because of whether it can cross that cell membrane or not. If it can cross the cell membrane, it's going to be steroid-based. If it cannot, it will be protein-based. So if we look at this, let's look at the example that we have here on the right here. So this one here is our G protein receptor. So that's what we're looking at here. Again, this is another time of an example that if you needed to write about it, you could pull this one out of your pocket because you got it down. Okay, so our example here is a G protein receptor. We're going to find these in eukaryotic cells. So what will happen with these receptors is the ligand is going to bind to the receptor. And when that ligand binds to the receptor, you can see here this GDP gets activated into GTP. And so the activation of the receptor causes the phosphorylation of the GDP into GTP. And so what that will do is that will activate this G protein. See how this orange protein here changed shape because structure and function. So it's inactive here in this kind of upside down shape. And then when it gets flipped over there, that G protein is active, but it only becomes active once the receptor binds with the ligand. So that G protein slides across the membrane here. You see your arrows here taking it this way across the membrane. So slides across the membrane, okay, and it then binds to the enzyme. The G protein binding to that enzyme is what's then going to trigger the pathway inside of the cell. And so I get this temporary change. As soon as that GTP is broken down back into GDP, that G protein will become inactive, head back over there. When it releases from the enzyme, that enzyme shuts down that signal transduction pathway. So I get a temporary change due to the signal that was on the outside. So the protein ligand bind to the receptor. That triggers the GDP to be phosphorylated into GTP, which activates the G protein. The active G protein slides across the membrane over to the enzyme, where it will then bind with the enzyme and activate the enzyme. The enzyme activation triggers our transduction pathway within the cell. And then again, when that G protein releases from the enzyme, that will all get turned off. So I get a temporary response, okay? And then as soon as that signal is gone, that response will shut down pretty quick. Another example is down here at the bottom, this ligand-gated channel. So again, I've got my messenger or my ligand. You can see it here in the green, coming in and bonding with my receptor. Again, ligand and receptor are matches. Okay, and so that's what allows the two to join together. That's what allows it to be specific. That causes the channel to open. You'll see on the right here, the channel is closed when there's nothing binding to the receptor. But when the messenger binds to the receptor, that opens up the channel. Okay, and that allows things to flow in. Okay, it allows ions to flow into the cell, out of the cell. But it will change, what it will do is it will change the concentration of the ions within the cell. If I'm at a neuron, when a neurotransmitter binds to a muscle cell or binds to another neuron, it binds to these ligand channels. The channels open up and sodium goes rushing into the cell. And that's how the cell knows that it's gotten a signal to either contract the muscle or to pass the message on. Okay? And so that it'll change the ion concentrations within the cytoplasm, which tells the cell that changes are coming and that it needs to change and that there's something has changed in the environment that it needs to respond to. So let's look at what happens once that signal has been triggered inside of the cell here. So here's your messenger right here, my ligand, right, bound to my receptor. I know it's a protein one because my receptor is on the cell membrane. This is that same G protein, right? So that activated the G protein. G protein is headed over there to the enzyme. And so now that activation of that enzyme has triggered that signal. So when we trigger that signal, those are called signaling cascades. And the purpose of the signaling cascade is to amplify the signal, to make the signal increase in strength enough so that the cell will have an appropriate response to it. Okay? And so those cascades are helping pass the signal from the receptor to the specific target areas of the cell. Like, for instance, maybe the cell needs to grow. Maybe it needs to secrete something in response to the signal. Maybe it needs to alter what genes are being expressed. And so those are all different areas of the cell. And so this signaling cascade helps get the message to the appropriate area so it can have the appropriate response. And a lot of these signaling cascades commonly use what we call second messengers. These are going to be molecules that are within the cell, and they're gonna be the ones that do the cascade. They're gonna be molecules that are going to relay and amplify the signal. And a very common example here is our cyclic AMP or CAMP. So CAMP is commonly found when the activated enzyme from that G protein receptor. And so that activated enzyme here, so this enzyme here has been activated, that'll form that cyclic AMP. And when it forms that cyclic AMP or CAMP, that's what's going to trigger all of these cascades, these signaling cascades. And these signaling cascades are usually based off of protein kinases. Protein kinases, in general, are going to be molecules that phosphorylate other molecules. So when we phosphorylate something, we add phosphate to it. And so that activation of the enzyme triggers the formation of the cyclic AMP. The cyclic AMP starts those cascades, starts those protein kinases going so that they will start phosphorylating other protein kinases, other molecules, moving that signal along so that it can get to the appropriate area of the cell for the right response. So the last part here is the actual cellular response. So we have the reception, we have the phosphorylation cascades that are happening in between, making sure that the information is actually getting to the area where the response is going to happen. And then we have the actual cellular response. And there's a variety of things that can happen with the response. You could alter gene expression. Most of our steroid or lipid-based ligands will alter gene expression because they'll go all the way into the nucleus, straight to the DNA and impact the DNA. You can alter the cellular function itself. You could tell the cell to start making something, to stop making something, to release something, to take something in. It can also change phenotype. Remember, phenotype is the physical expression of the proteins. It could also trigger apoptosis. Apoptosis is basically cellular suicide. So the information could be telling the cell that it needs to go ahead and have a programmed cell death for whatever reason it was happening and whatever's going on in the environment. So like before, we're going to look at some more examples so you can kind of see this in action in a variety of ways. So we're going to look at 2 examples here with cellular response. The first one here is the epinephrine example that's happening there on the right. Okay, yes, that is a crazy diagram. There's a lot going on. There is no way that you're going to be expected to memorize that diagram. What you need to get down is my possibilities for response, and I need to be able to adequately talk about them. So we're going to use an example to talk. We're going to use a couple of examples to talk about that. So looking at this epinephrine example. So epinephrine is adrenaline. So epinephrine is released from the adrenal glands. And so that epinephrine is a hormone. This is an endocrine communication. So it's released from the adrenal glands. It's traveling in the bloodstream. And one of the responses it causes is on liver and skeletal muscle cells. Okay, so here's my epinephrine here. Okay, and so on these liver and skeletal muscle cells, it'll bind to these G protein receptors. So there's my reception there. And that's going to activate the adenyl cyclase enzyme. So it's going to activate that enzyme. You can see, here's my enzyme right here, right? Here's my binding to the receptor. Here's the activation of my G protein. That activates the enzyme. Here's my second messenger. There's my cyclic AMP. So now I've produced this cyclic AMP. Cyclic AMP activates those protein kinases. So here I get my phosphorylation cascade happening. Okay, I'm amplifying my signal. Okay, and so I could be activating multiple protein kinases here so that I can, spread the word faster. I can spread it stronger. But I've got that cascade happening, that signaling pathway happening. And at the end of that pathway, I finally get my response here. And the final product at the end of that pathway is this enzyme here, this glycogen phosphorylase. So we've gone through multiple phosphorylation kinases here. Okay? And I end up with this enzyme as my final product, which is, again, glycogen phosphorylase. And so what glycogen phosphorylase does is it takes glycogen and it breaks it down. Okay? It takes glycogen, it breaks it down into glucose. So now these liver and skeletal muscles that have been storing glucose, they store it in the form of glycogen, so long chains of glucose. They can now release this broken down glycogen, so now these little pieces of glucose from the glycogen getting broken down, they can release that out into the bloodstream so your cells can have better access to glucose to do more cellular respiration. Because you've released adrenaline, which means you're ready to fight or flight, which means you need things functioning at optimal abilities. So in this instance, as I look at my 3 parts of my pathway, my reception is the epinephrine binding to the receptor. My phosphorylation cascade, or my signaling cascade, my transduction pathway, is happening here in the middle here, okay, from that cyclic AMP activation through all of those phosphorylation cascades. And then my cellular response then is to break down the glycogen and release those glucose pieces now out into the bloodstream. So my response was I altered cellular function by producing this enzyme that's making this glycogen now. Not making glycogen, releasing glycogen, breaking down the glycogen to release the glucose. So let's look at this example. So this is another example here, and this is happening in plants. And we're looking at ethylene. So ethylene is a plant hormone. Ethylene is the plant hormone that causes fruits to ripen. Okay? If you've ever had, like your grandma or your mom, has fruit that's not quite ripe and puts it in a paper bag on the counter, what they're doing is they're trapping that ethylene gas, and that's helping encourage the ripening. How we get, one of the ways that we get fruit from, we get apples are only really ripe in the fall. But we have apples in the grocery store year round. Well, those apples went to the warehouse and they were stored in the warehouse. They were picked early, stored in the warehouse. And as those apples sat there, all of that ethylene was pumped out. No ethylene was able to get to these apples. And so as it's time to ship those apples out so they can be shipped out periodically year round, they pump the ethylene back in so they can finish the ripening process. Okay, so ethylene is a hormone that's released from plants. It's released in the form of a gas, okay, and it'll travel through the air. And so that's how one ripe banana ripens the whole bunch. So it's a plant hormone that's in a form of a gas, so it can travel through the air, and it can pass through the cell walls of plants. And so the ethylene receptor is, the ethylene will bind to the receptor on the membrane, and that's going to trigger our transduction pathway. Again, you don't need to memorize this. All right, so it triggers my transduction pathway. You can see I've got a kinase cascade happening, all these various arrows here. Okay, that's showing that phosphorylation happening. And what will happen is that transduction pathway, you'll notice over here, this is DNA. So that transduction pathway triggers what we call transcription factors. Transcription factors encourage transcription. Remember, transcription is part of protein synthesis, right? Gene expression. Right, we go from DNA to messenger RNA with the transcription, and then messenger RNA to the actual protein, the translation. So this transduction pathway ends up at these transcription factors, which are going to affect gene expression. And so what it will do is it will turn on genes that were previously turned off. So we'll turn on genes that help soften and break down the cell wall. That's part, if you think about it, that's part of the ripening process of the fruit, right? It softens, it gets a little bit sweeter. So it's turning on the genes that start breaking down the cell wall and converting those stored starches into the simple sugars, breaking them down into those simple sugars so the fruit gets sweeter. And so that's all going to happen. Again, as the environment changes, that triggers the release of the ethylene. The ethylene binds to the receptor, triggers my pathway. My pathway ends up affecting my DNA, so my gene expression. So this time my cellular response is an altering of gene expression, okay, which will then end up as, for the organisms as a whole, ripening the fruit. So the last thing we're going to look at here is, what if there are changes in the signal transduction pathway? How does that affect things, and what happens here? So if, let's say there's a mutation in any protein along the pathway, it could be in the receptor, because those could be proteins. Okay? It could be along those cascade, those protein kinases along the cascade. Any kind of mutation along the way, okay, that can cause the downstream components of this pathway to malfunction. For example, look over here at these rasopathies. Okay, so what's happening here, okay, is when there is a problem within these proteins. So you can see some examples of these down here at the bottom. Okay, various syndromes, various problems where the proteins are malfunctioning. Okay, that can trigger everything that comes along behind it, can be messed up. Okay, and so what you have over here on the right with these little like lightning bolts and the dark blue boxes next to them, those are possible therapies that could help with this. Like, for example, the MEK inhibitors. So if I'm causing too much of this cell growth, I may need to shut those down because of the, so I have the mutation, and the mutation is causing too much of the cell growth, and so I need to start shutting them down. Okay? And it's important to regulate this, because we've got, if you think back to freshman year, mitosis, cell cycle, remember, as the cell divides, it gets signals as to whether it's time to divide or not, or there's space, or there's resources. And these potential mutations here along pathways, they can lead to cancer. You can get things that affect the regulation of the cell cycle. Like right here, I've got overgrowth, proliferation, cells surviving when they shouldn't be surviving. And so any kind of changes in these signal transduction pathways, they can lead to serious consequences for the organism. And so you'd have to kind of break down where these changes are happening to try to target the therapy. Okay, looking at this example here with chemical interference. So I could have interference in the pathway by natural mutation, okay, or whatever has caused that mutation. But I could also have like man-made interference with chemical interference. And chemical interference can interfere any component, again, along the pathway. I could have something that competes for the receptor. I have something that interferes with the phosphorylation cascades. Chemical that interferes with the actual response. And they can work both ways. They can either activate a pathway or it can inhibit a pathway. So this is not strictly interfering in a bad way. They could be trying to activate something to get it going. Okay, so the example we have here is muscle relaxants. Okay, muscle relaxants will block acetylcholine. Acetylcholine is the ACH that you see in the diagram here. Acetylcholine is the neurotransmitter that is released from a neuron across the synaptic cleft to bind with the muscle, to tell the muscle that it's time to contract. And so what muscle relaxants will do is they will block acetylcholine. So acetylcholine, you can see how the muscle relaxers are in the way. Okay? They will block the acetylcholine's ability to bind to the receptor. So now I've interfered with the signal transduction pathway by blocking the reception. So if there's no reception, then the ligand channels don't open. And if the ligand channels don't open, then it doesn't allow for sodium to come into the muscle. If no sodium ever comes into the muscle then doesn't get the information that it needs to be contracting. So if these receptors keep this, if these muscle relaxants keep that channel blocked and closed, then there's no sodium rushing into the muscle, and so the muscle doesn't contract, and so the muscle will stay nice and relaxed. That's why if you've been on muscle relaxers or you've seen anybody on muscle relaxers, they don't really have full control, even if they want to. Even if they send the signal that tells it to contract, they're still not getting the response that they want because the medicine is chemically interfering with the signal transduction pathway, in particular the reception part of the pathway. Okay, so once again, I know you got a lot of examples to try to help explain these things. Pay attention to the examples. Get the examples down. If you can own and understand the example, then you can take that and you can apply it to all different kinds of examples. Okay, so we'll work with these some more in class, okay, and just keep, plugging away at this.